Hi everyone, it's my great pleasure to share our recent work to the symposium, which is the focus ultrasound-mediated intranasal delivery of AAV to targeted brain region with minimal systemic exposure. The majority of the neurology diseases are caused by the dysfunction of single gene or small number of pathogenetic proteins. Gene therapy is using the nucleic acid for treating such kind of disease. The nucleic acid can be packaged into the AAV, adeno-associated virus. Adeno-associated virus or AAV is the most used gene carrier. Once delivered to brain, it can treat the brain disease either by express the normal gene and to produce the healthy proteins. It can also silence the mutated RNA or correct the mutant gene by using some genetic editing tools. However, the delivery of the AAV to brain is challenging, which potentially limited the development of gene therapy for treating the brain disease. The current most used techniques are the direct injection or the intravenous injection. However, direct injection is invasive. It sometimes requires draining multiple holes to the brain. And also the delivery from direct injection is very localized, which can cover only a small region of brain, which is not really applicable for the brain disease that affecting the whole brain. The intravenous injection is non-invasive and can cover the global of our brain. However, consider AAV is relatively 15 to 30 nanometer, it cannot cross the blood-brain barrier. So intravenous injection of AAV requires special design of the capsid to cross the blood-brain barrier. Also, the AAV will go through the blood circulation to the whole body, which may induce the risk of systemic toxicity. And indeed, the hepatic toxicity of AAV is one of the biggest concerns in the AAV-based gene therapy. So the advanced method for AAV delivery are required for safe and effective gene therapy to treat the brain disease. We propose to use intranasal delivery of AAV, directly deliver the AAV from the nose to the brain, which can bypass the blood-brain barrier without entering the blood circulation. As shown here, intranasal delivered agent will travel along the perivascular space, which is outside of the blood vessel. We also propose to use focus ultrasound to further locally enhance the delivery efficiency of AAV to the focus ultrasound targeted brain region. So the objective of our study is to evaluate the potential of fusion focus ultrasound enhanced intranasal delivery for efficient delivery of AAV to mouse brain with low systemic biodistribution. In the experimental part, we used the AAV5 encoded EGIP protein to test our hypothesis. This AAV was the first intranasal delivered, followed by the focus ultrasound targeting at a superficial brain region, the cortex, and also a deep brain region, brainstem. 1 month after the AAV delivery, we sacrificed the mouse and used both the fluorescence imaging and PCR to visualize to see the delivery outcome in terms of the brain delivery efficiency and the biodistribution. We also added direct injection, intranasal delivery, and the focus ultrasound combined with microbubble-induced blood-brain barrier disruption to compare with this fusion delivery. And for the direct injection and fast BVD, the targeting is also at the cortex. Now let's look at the result. First, we can look at the fusion delivered mice. The EGIP fluorescence signal observed in the targeted cortex region indicated that AAV EGIP was successful delivered by fusion and subsequently expressed the carrier gene. The direct injection also showed successful gene delivery and expression, while both in the intranasal delivery and the FUS-BBD delivery group, we have not found observable EGIP gene expression. The PCR quantification of the genomic DNA of EGIP is consistent with the fluorescence image observation from the mouse brain. So from this part, we showed that fusion can successful and efficient deliver the AAV to the targeted cortex. And next, we want to evaluate the biodistribution of the AAV after delivery. So to evaluate the biodistribution, we collect the major organs from each delivery group, including the fusion, FUS-BBD, and direct injection. We processed the organs and used the ddPCR to quantify the EGIP transgene copies in the organs. We found that EGIP transgene accumulation in most organs didn't significantly differ after fusion delivery and direct injection, but besides lung. We also found that fusion and direct injection group were all associated with much lower EGIP transgene copies than the FUS-BBD delivery. That might be because the FUS-BBD delivery in this group, the AAV was intravenously injected and traveled in the blood circulation, so it resulted in higher accumulation of AAV in most organs. In the fusion group, so we found the AAV accumulation is higher in lung. That also might be because we used the AAV5 serotype, which is reported that can be easy better to transduce the airway. Besides showing that fusion successfully delivered the AAV to the cortex with minimal systemic exposure, we also performed the fusion delivery of AAV to the deep brain region, which is the brainstem. This is to confirm that fusion has the potential as a platform technique that can non-invasively deliver the AAV to different brain regions. The strong EGIP fluorescence signal at the brainstem indicating the successful targeted delivery of AAV by the fusion. Consider the EGIP protein can be transported in brain through the nerves. We also confirmed the EGIP gene delivery and transaction by directly detecting the EGIP on RNA using the RNA scope insult hybridization assay. This result demonstrates that AAV transduced cell was confined within the FUS targeted brainstem region. We quantified the fluorescence intensity of both the EGIP protein and EGIP RNA in different brain regions. And the quantification is consistent with the fluorescence observation. We consistently found the fluorescence intensity is higher in the brainstem region, including the midbrain, pons, and medulla. We also found some fluorescence. We also found higher fluorescence in the cerebellum. That might be because our ultrasound beam is kind of long, so it can cover the whole areas along the ultrasound beam. So in the future study, we probably can also reduce the size of the ultrasound beam to achieve a better localized delivery. So in conclusion, in our study, we found that FUS with intranasal delivery, which is fusion, can achieve efficient AAV delivery and gene transduction at the focus ultrasound targeted brain region. We also found the fusion delivery of AAV can minimize the systemic exposure of AAV to other major organs besides the lung. And finding from this study suggests that fusion is a promising technology for AAV delivery to brain, with the potential to be translated to clinic for the treatment of multiple central nervous system disease. And in the end, I would like to thank all the Chen's Ultrasound Lab, my advisor Hong Chen, and all the lab members for their help in the research and also the funding support. Thanks for your attention, and I'm happy to answer any questions.